This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. It's time for TWiM, This Week in Microbiology. And today is February 9th, 2016. Hi, everybody. I'm Vincent Racaniello, and you are listening to the podcast that explores unseen life on Earth. I am coming to you today from just outside of Washington, D.C. I'm at the American Society for Microbiology Biodefense and Emerging Pathogens Research Meeting, and I've grabbed two of the participants in this meeting, and we're going to chat with them about their work. And I just need to tell you, before I introduce them, that the weather's miserable today. It's cold and it's raining. On the way down it was snowing, and as I came further south it turned to rain. And let's leave it at that. Not a very pretty day. My two guests here from the meeting, all the way to my right, from the University of Colorado in Fort Collins, Rebekah Kading, welcome.
Thank you.
And on my right, from Northwestern University Medical Center, Wyndham Lathem. Thank you for coming.
Hi, great to be here.
Thanks to both of you for taking time out to join us. We're going to chat about what you guys do. And I want to remind the audience, if something doesn't make sense, there's a microphone here. You can come up and ask us a question, if you will. We'll see who's brave enough to do that. All right. First, I want to find out where you guys are from. Rebekah, you're now in Fort Collins, but are you from there originally?
I'm from Delaware originally.
And where did you go to college?
I did my bachelor's at the University of Delaware in entomology. My master's is from the University of Arkansas in entomology, and my PhD is from Johns Hopkins.
And at Hopkins, what did you work on?
I worked on malaria.
Malaria. Mm-hmm. So from a very early age, you were interested in bugs.
They kind of hooked me in my undergraduate days, yes.
Is there any reason why you got hooked on bugs?
I was interested in wildlife conservation, and at Delaware at the time, they didn't have that as its own major, so I was forced into taking the entomology classes. Yes, but then it grew on me very quickly, and so I'm glad I went down that path.
Yeah, from that came a career studying microbes that bugs carry around.
It served me very well to have a solid background in it.
So you've been in Fort Collins, not for very long, right?
I moved out there in 2007 and did my postdoctoral work with the CDC, but I've only been at Colorado State University about a month now.
So, we had an ASV meeting there a couple of years ago.
Yes.
Did you go to that?
I was not able to attend that.
All right, we're coming back in a few years.
Yes.
So you can come next time. There are lots of virologists there. You might be interested in going. Wyndham, where are you from?
So I was actually born in London, England, but I grew up in Connecticut, just outside New York City.
You have no accent whatsoever.
No, I guess not. Well, I only was in London for about 2 weeks. So just enough time for my mom to pop me out, and then to Connecticut.
You grew up in Connecticut?
I did, yep.
And college, where was that?
So I went to college in the mid-Hudson Valley of New York at Vassar College, so a small liberal arts college. It's where I also got my first exposure to microbiology. I worked in a research lab there studying Neisseria gonorrhea. So,
do you remember who that was?
Yeah, so there's a professor there. She's since retired, Penina Norad. And so it's a little different, and certainly now working at a medical center where we can do infectious disease research, working on a pathogen in college, not being able to actually do pathogenic studies. Sure. So we worked on some biochemistry, but it was my first exposure to that. Yeah.
And then for a PhD, where did you go?
So actually I took a couple of years off and worked at Rockefeller University as a lab technician. And from there, I went to the University of Wisconsin in Madison.
Whose tech were you at Rock?
In Jim Darnell's lab.
Wow.
Jim Darnell Senior, not junior.
Right. Yeah. And what years were they?
So that was from 1996 to 1998.
All right, so we were in the city together. I never saw you.
Yeah, well, it's a big city.
All right, and that's a great lab to have a experience.
No, I learned a tremendous amount of just not just techniques, but also about thinking about science and how to to ask the right questions and troubleshoot and all those sort of things. Yeah.
And who did you work with? This PhD at Wisconsin.
Yeah, that's right. So I worked in the medical microbiology and immunology department with Rod Welch, who's now the chair of that department, and so I worked on enterohemorrhagic E. coli.
And postdoc?
So then I went to Washington University in St. Louis to work with Bill Goldman, who was my primary advisor, and then also his wife Virginia Miller as a co-advisor. And we actually started the plague project in those groups, studying pneumonic plague there.
All right, so that's where the plague, your interest in the plague.
That's right, been doing it ever since.
It's been plaguing you ever since.
It's funny because when people say, oh, I avoid that like the plague, I think, oh, then you must do it all the time.
Yeah. So that's one of the things we're going to talk about today, your interest in the plague. And that of course, can be transmitted by ticks. But, Rebekah, your interest is mosquitoes, not ticks.
Mosquito-borne viruses, yes.
So let's talk a little bit about Rift Valley fever virus.
Sure.
You're having a session on this today, right?
Yes, this afternoon we're having a session on
And let's talk a little bit about it, when it was first found, what does it do, where is it, and so forth.
Well, it's an East African virus. It was originally discovered in the 30s, I believe, and it's not only is it a potentially important virus for public health, but it's a veterinary pathogen as well, which makes it scary on that level too. And that it's also a select agent, and so it's one of those viruses that's been on my radar and the radar of my colleagues for quite some time, and it's one that we're keeping an eye on, and I don't think we're quite prepared enough for that one to come here, and I think we need to be proactive, especially with Zika virus knocking at the door right now.
We keep getting reminded that we're never ready. Right.
Right.
Whenever, but even for viruses that we know of,
right.
we often are not ready. So
it's for the next emerging arbovirus. We may not know what it is, but in this case, I think we have some heads up as far as well. Here's one that it showed up in Saudi Arabia in 2000. If it can, this is a virus that's closely tied to rainfall events. If it can emerge in Saudi Arabia, it can emerge anywhere, and so I think we need to be ready for it.
So the majority of the cases are in Africa still?
Yes, it tends to cause outbreaks every 10 to 15 years or so, and very closely tied to coinciding with significant excess rainfall.
Okay.
The mosquitoes that transmit it are emerging from areas that flood, and that usually starts the epidemic.
And the animals that get infected include cows?
Cows,
right?
sheep, goats, livestock.
So these are peri-domestic animals,
yes.
Right? And it kills them?
It has up to 100% mortality rates in neonatal animals especially. It causes abortions in livestock.
And so is there a reservoir in nature that harbors the virus as well? Do we know?
To the best of our knowledge, the virus is maintained by vertical transmission in the mosquito, but there's more research to be done in that area. But there is some evidence that the virus is hanging out in the eggs of these mosquitoes, and then when they flood, the eggs hatch and the mosquitoes are already infected. But as far as the dynamics of interepidemic transmission, there's still a lot of work to be done.
So you think it exists solely in mosquitoes and not any other mammalian species? I
think there's evidence for low levels of transmission going on elsewhere, right. but as far as the smoking gun, a reservoir for Rift, I mean, people develop viremias that are high enough, and most of the cases are not the hemorrhagic fever cases that we hear about. So I think that there could be low levels of transmission going on in between the big epizootics that we hear about.
So when there's an outbreak, it typically involves farm animals, right?
Yes, domestic animals and people as well.
So do the animals serve as amplifying reservoirs for the virus? Right?
Yes. And people too. So the transmission can occur through the mosquito bites, mosquitoes biting people, mosquitoes biting animals. It's the difference between Rift and West Nile. For West Nile virus, people don't develop high enough viremias to infect mosquitoes, but for Rift we would. And so the amplifying host can be people, they can be large livestock animals, but then people can also become infected by directly handling infected animal tissues. And so
you don't have to be bitten.
Correct.
So if you have a farm and your animals are infected, you could be infected by mosquitoes, which bite you and give you a virus, or by just handling the animals. either way?
Infected animal
animals.
tissue.
So when a human is infected, what's the outcome?
It's there's a lot of different pathologies that could be febrile, but then there's an ocular pathology, there's neuropathology, there's the hemorrhagic disease, and so it can be pretty severe.
So what's the case fatality rate, roughly?
Case fatality rate, I think it's a fairly low percentage that would develop the serious illness.
Whereas with your Yersinia, it can approach 100%.
It can,
right?
Yeah.
Okay.
Yeah.
So if a human gets sick, is there anything you can do? There are probably no antivirals or vaccines, right?
There is a vaccine strain, but it's not a commercially available vaccine. There is veterinary vaccines in development as well.
Okay, now it has spread outside of Africa, right? It's gone to Saudi Arabia.
The Saudi Arabia epidemic. Mm-hmm.
Has it come to
at
Europe?
At this point in time, that's what we're looking at.
So that's what your concern is.
But it has the potential to leave Africa. It's shown us that, and so I think it's just a matter of the virus, the host, the mosquito, the right person getting on an airplane infected. It's just it has the potential to jump. It's just all those factors have to coincide at the right time.
I understand that the virus can infect many different kinds of mosquitoes. Is that right?
That's correct. Mm-hmm.
So and we have some of them here in the U.S.?
We have very competent vectors for Rift here in the United States.
And so actually in one of your papers you talk about mosquitoes taking plane rides. And I thought, people talk about it a lot, but there's actually evidence that it happens, right?
Yes, it's conceivable, yes, and it has happened. That would be the big question now is if Rift were to come here, would it be through an infected mosquito taking a plane ride, or would it be through an infected person taking a plane ride? I think the quarantine things that we have in place for infected livestock, it wouldn't happen that way, but mosquito or person would be my bet.
Maybe it's already been in the U.S., but we just didn't know it. Is that possible?
That's possible.
I mean, do we survey enough?
Again, I think that that a lot of the cases, there might be someone that is infected but comes back and nothing happens because the environmental conditions weren't right. It was in the middle of winter, so there was no mosquito to bite that person, or things all have to the stars have to align for this sorts of things to happen.
You have to have a perfect storm.
Correct.
Like we did in '99 when West Nile virus.
That's right.
Came to New York City.
That's right.
It was August, right? Which was perfect. Lots of mosquitoes around. It was very dry. And that took off.
It did. And an important point about that to make also is West Nile virus is a zoonosis. So it got into the birds. It's a bird virus, and so so it established itself here that way. Rift is also a zoonosis, and so that something like yellow fever, chikungunya, dengue, maybe there's a sylvatic cycle, but for a large part of the transmission is in urban centers between people and mosquitoes. Right. You can break the transmission cycle. We used to have malaria, yellow fever here in the United States, but now we've got air conditioning and televisions, and so people are inside. They're not exposed to mosquitoes the way we once were. But that's not the case with these zoonoses, and so the birds harboring West Nile virus are not inside watching TV, and so they're getting exposed to mosquitoes, and I think the livestock infected with Rift would be the same way.
I just want to ask: is there any evidence of person-to-person transmission
for Rift?
Yeah.
Not that I'm aware of.
Okay.
So, what would you need to go person to person? I mean, mosquito would have to pick up enough virus in a blood meal to be able to transmit, right? That's the minimum.
Right. And so for mosquito then that brings up the issue of mechanical transmission versus biological transmission. So when a mosquito takes in an infected blood meal, that virus has to get out of the midgut, through the mosquito, into the salivary glands before it can be transmitted, and that takes time. In Rift, it can be just as short as a couple of days, though. It can be pretty quick, but it still has to go through that process. But then there's mechanical transmission, where that would be just virus particles stuck to the mosquito's mouth part, or a biting fly, for that matter. Stable flies, something that would be a very messy feeder that could mechanically move virus particles from one vertebrate to the next.
So one of the things you look at is vector competence.
That's correct.
Maybe you could tell us how that works. What are you looking for? What do you do?
So vector competence is the biological ability of the arthropod to transmit. And so there's the barriers to transmission that I just described. So there's the midgut infection barrier. So the virus has to be able to infect the midgut. There's the midgut escape barrier. So once it gets in the midgut, it has to be able to replicate and escape from the midgut. And then there's the salivary gland barrier. So once it's in the body of the mosquito, it has to be able to infect the salivary glands to be transmitted. And this varies by mosquito species. So some mosquito species can become infected, but the virus can't get out of the midgut. Sometimes it can get out of the midgut, but then it can't get in the salivary glands. And so there's variation in the permissiveness of the mosquito to be able to harbor virus replication. There's also variability within a species. And so sometimes one population in the United States of a particular species will be very competent, and another population will not. And so there's no easy answer. And then, there's other factors that weigh in. The environmental temperature at the time can affect the speed at which all of this happens. The presence of other organisms, such as microfilaria or parasites, can punch a hole in the mosquito midgut, because mosquitoes are not out there flying on and feeding on laboratory animals. They're feeding on animals that are infected with other things. And so this can help the virus bypass those midgut infection and escape barriers. So there's a lot of different factors to consider.
So how do you measure these? Do you take the mosquitoes in the laboratory and feed them virus, or do you catch them and look?
So in the laboratory, then you would feed the mosquitoes either on a viremic animal or give them an artificial blood meal with a known concentration of virus. You would hold them for say a week or so, and then you can look for infection rates and dissemination rates. So infection rates. So what I would do is take all the legs off. And so if we just...
Wait, did you say you take all the legs off?
Take all the legs off of the mosquito.
Why do you do that so it won't...
So that would be an indicator of the virus disseminating. So if you find virus in the legs of the mosquito, that means the virus has gotten out of the midgut. And so an infected mosquito body would indicate an infection. Infected mosquito legs indicated dissemination.
So it's not just for sadistic reasons. You're plucking off legs.
And so then there's different ways to measure transmission as well. So you can either have that mosquito feed on an uninfected animal to see if that animal gets sick, or I use capillary tubes and have them insert the mosquito's proboscis into the capillary tube and then measure to see if virus has been expelled in the saliva.
So in your lab, how many different kinds of mosquitoes do you have to do these experiments with?
I think... Well, then a lot of this work has been done by Mike Terrell at USAMRIID, and he was bringing mosquitoes in from the field, and so that would be... so there are laboratory strains we can look at, but it's important then to look at the genetics of mosquito that's more closely removed from the field population, because laboratory mosquitoes are laboratory mice in some cases. They're not replicated what's happening in the field.
So is a mosquito in Colorado of a species the same as the same species in Africa, or are there going to be differences?
No, and there's different. There's one of the major vectors of rift in East Africa. We don't even have that subgenus here, and so the ecology of how rift might be transmitted here will be completely different than what's in Africa, and that's what we have to understand.
I think it's important to point out that Aedes aegypti here is not the same as it elsewhere, right? But everyone assumes that it is.
Right.
We have the vector, therefore the virus is going to spread. But that's not correct.
Not necessarily.
And you're the one who figures that out or not? You do the experiments that need to be done to answer these questions.
That's what the research I like to do, yes.
So
important questions.
For, what do you call it, Rift?
Rift.
Rift Valley Fever Virus, RVFV. Okay, Rift Valley. What mosquitoes can carry it? What genera?
It's the predominant genera in Africa is Aedes, but there's Culex some Culex species also. But the important part is which mosquito species are going to be feeding on livestock and people and large mammals. And so here in the United States, some that we're worried about again are in the genus Aedes that feed on large mammals. Aedes vexans would be one, but this is one of those species where the vector competence is different in different populations of the United States. So Aedes vexans in Florida, Louisiana, are very competent for rift. Aedes vexans in California, Colorado, not so much. And so, but in general, that's a very abundant mosquito, feeds voraciously on humans and other large mammals. It's competent for rift. So that's one. Aedes japonicus is another one, very competent in the laboratory. This is a mosquito that's introduced to the United States. So it's one that to keep an eye on and do some more work on. It's predominantly on the East Coast. One that I'm interested in is Culex tarsalis. Culex tarsalis, important vector of West Nile virus. What role it would play in rift transmission is unclear yet because it does feed a lot on birds, and birds are not competent hosts of rift. But Culex tarsalis, in the late summertime when some of its favorite bird species are dispersing after their breeding season, then shifts and feeds more on dogs and cattle.
So, in New York City, we don't have cows. We don't have sheep, as far as I know. So would Rift Valley not be a problem for us New Yorkers?
There's deer, and we don't really know what deer are going to do, and if deer are competent. And there's certainly deer overpopulation in the Northeast is a problem, and there are mosquitoes that feed on deer and feed on people. So that's a huge unknown that needs to be addressed.
So it could. You don't know that deer can be infected with the virus. Is that something you're trying to figure out?
That experiment needs to be done, and that's a difficult one to be done. But yeah, that's a black box.
I think my parents have a lot of deer running through their backyard all the time. I'm sure they'd be happy to let you have some of them.
I can have some, okay.
Yeah, we have, as you said, we have too many deer, and they periodically have culling where people are allowed to go out and shoot them. And you can just think about it, the northeast of the U.S., people are just going out shooting deer. It's not a good idea. It's not a good plan. Maybe we need to get them another way, like with a virus. No, we know that doesn't work. We know that doesn't work. So why is Rift Valley virus a select agent? What could be done with it?
It's a Rift Valley fever virus. It has the potential to be aerosolized and weaponized, and so that would be a reason. But it also can cause very severe pathology. There's not a human vaccine readily available, and so just the potential for this virus in a number of different forms.
So it's not that we're not worried about the animals. We're worried about people.
Well, it's a USDA listed select agent as well. So it's also it's a crossover. It's human and veterinary.
Because it could be destructive to
very, very much
farms that grow animals, right?
Public health and for the veterinary sector and for the economy too, for that matter.
What tier of select agent is it?
I don't believe it's tier 1. So
But it is a select agent.
It is.
So what is being done? You mentioned earlier we have to be prepared for the eventuality if the virus would arrive. What's being done?
Well, I'm hopefully at CSU we'll be starting up a research program to investigate some more of the entomological questions. We don't know anything about the ability of rift to be vertically transmitted by North American mosquitoes, and so that's one question. Or for if it could potentially overwinter in adult mosquitoes that are hibernating. So those sorts of questions of how it would maintain itself in nature. Mosquito control is a very species-specific targeted intervention type of activity. So we have to fully understand what species of mosquitoes are transmitting, would be transmitting rift in order to design what control measures we would go in and control it from that perspective. And then so there's a ton of work left to be done on the entomology side. The vaccine development side, there's more to be done. And then, coming up with a preparedness plan.
Does that include a vaccine and antivirals, or is it too soon for that?
I think we should be moving in that direction. Absolutely. Yeah. I think we have a lot more to do with Rift, but we're ahead of where we are with Zika virus, where we are with Rift right now. So we have a head start.
Yeah, it's always hard to know what to focus on because there are lots of zoonotic viruses out there.
Right.
You can't do all of them.
Right.
And we got caught with Ebola. We got caught with Zika. And you probably feel you're fighting a battle that can't be won because you're a sole voice. But it could be in 5 years there's an outbreak of RIF in this country, and then we'd be caught again. What do? You can't work on everything, right?
Right. It's a struggle, and a lot of times we're very reactionary in what we need to do, and it's hard to find the funding to be proactive when something's not a problem yet. And so so it's a struggle, but I think we just keep plugging forward, and I think with Rift, it's already jumped once to Saudi Arabia, so it's shown the potential.
Probably doesn't hurt that outbreaks raise awareness in the public, right? So you have Ebola last year, and then Zika this year, and then you can go out and say, look, this is why we need more research and development on these viruses. Do you think that's of any use?
Yeah, I guess that education is an important component of it as well, without causing mass panic. But just being informed and being aware, and protect yourself against mosquito bites. Dump standing water containers in your front yard. Those kinds of messages are not expensive to convey.
I wanted to ask you, so mosquito control measures can make an impact, can't they?
Absolutely.
Like you said, getting rid of standing water.
Yes.
Some spraying as well. Many localities in the U.S. do spraying in the summer, right? And being sensible about your dress and so forth. So mosquitoes bite all the time, though, right? Day and night?
It depends on the species. Species Aedes aegypti, Aedes albopictus, Aedes vexans, they're day biters. But then Culex are more dusk and dawn. And so it depends on the environment that you're going to be in and what time of day you should be most protected.
Right.
But just, it's kind of a conscious decision in your lifestyle. My son plays baseball and in the evenings, and there are mosquitoes out, so we just have a can of DEET in his bag of gear, and just that's what you do.
So you've been asked this, I'm sure, a lot. What do you think is the potential for Zika virus to spread, say, like West Nile did in the U.S.?
I think, then it would be the zoonosis versus anthraprenosis. So I guess with Zika so far it's human mosquito, and so with a very urbanized peridomestic species, Aedes aegypti, and Aedes aegypti has a limited range in the United States. It's a very tropical mosquito, but there's big questions remaining, such as Aedes albopictus. What role is that mosquito going to play in more temperate areas? It has a much more plastic feeding behavior. So it could dilute out its effects, but it could be important in some locations, and so there's a lot of questions we don't know. And will Zika? I mean, this is Zika in the Zika forest in Africa is maintained by canopy biting Aedes in non-human primates, and so we don't know what, if any, vertebrate species here might be important as a reservoir for Zika.
I mean, for similar reasons, dengue and chick have not really taken off, even though they come to the U.S. It's person to person largely, right? And as far as we know, no animal amplifying host, and that probably limits spread, right? Whereas West Nile, as you said, goes into birds,
right,
among other things. It's interesting. I go on social media a lot and answer people's questions, and I always say, we don't know, we need to learn more, as you've just been saying, and people get very angry. Say, why do you say you always need to know more? Why don't you know everything? And I think that's a good point, that we always have a lot more to learn. We don't know every answer to the question, and if you want to solve things, you have to keep doing work. You have to support research. I wanted to ask you about a cool paper you published, which is right in my neighborhood, New York City. You studied this mosquito species that lives in the 91st Street subway.
Right.
This is the greatest paper I've seen, because apparently there's a population in that subway.
Yes.
Tell us a little about that.
Well, Laura Harrington from Cornell is the one who crawled down in the sewer and actually got those mosquitoes. But yes, that's a subtype of Culex pipiens called Culex pipiens molestis. And molestis is a form of pipiens that does feed more on mammals. It's autogenous, so it can lay eggs without taking a blood meal. and it can breed in very confined spaces. So all of these ecological characteristics, enable it to inhabit, areas like subway systems, and where it's very different. And so because of that mammal biting behavior of the molestus form, it's hypothesized that this is the genetic component of pipiens that's allowing it to be important for transmitting West Nile to people.
So these mosquitoes live all year round in the subway? Mm-hmm. And they bite rats and so forth that are down there as well as humans?
I would guess so, but then they don't have to take a blood meal to lay a batch of eggs. And so so the questions are, what sort of mixing is happening between above ground and below ground populations? Mm-hmm. How diversified are they? And then there's the Culex pipiens complex population genetics is a hot topic in entomology, and a controversial one, and complicated.
Yeah. So in the winter, these mosquitoes are no longer active in the subway, I presume?
Culex pipiens does go into diapause during the winter time. Okay.
And so this is not unusual to have populations of very specific mosquitoes in very specific places like that, or is it just a place where you found them and you study them?
That's that would my colleague Laura Harrington found them there.
So your colleague sampled them,
and
went
so
down to the subway.
an interesting way that here's the molestus population in North America. The famous one would be in the London Underground, studied there in the UK.
So it's not uncommon for mosquitoes to inhabit these man-made structures like subways and tunnels, sewers, and so forth. And it's a good place to sample them. I thought that was a cool story. Well, let's move on to ticks.
Fleas.
I get them confused. They're both tiny. Fleas.
You don't want them biting you either way.
I don't want either one biting me. Fleas. And your bacterium, Yersinia, which you grew fond of in Wisconsin?
St. Louis.
Mm-hmm. Bill Goldman.
That's right.
Right. Yep. And I want to talk a little bit about some work you've done to figure out where it came from, what made it so virulent. But give us a little history. How long have we known about? So Yersinia causes plague.
That's right. Yersinia pestis causes plague.
And there are a couple of different forms, right?
Sure. So there are several different forms of plague that can develop, all caused by the exact same species, same isolate. It just depends on how the bacteria are introduced into the body. Mm-hmm. So the main way is by being bitten from an infected flea. And so, of course, this is another example of an arthropod-borne pathogen. Mm-hmm. But instead of being biologically transmitted, I guess as you described it, having to go through the salivary system, pestis is mechanically transmitted by fleas. And so what happens is that when an uninfected flea takes a blood meal, the bacteria end up colonizing a valve-like chamber between the esophagus and the midgut. It's called the proventriculus. And so there's all these little spikes in there, and the bacteria adhere to that, and they grow to very high levels and form a biofilm. So that essentially blocks the flea so it can't get blood into the stomach. And so what ends up happening is it undergoes a very aggressive feeding behavior because it's trying to feed. So it bites and bites and bites. And what ends up happening is mechanical transmission. You get regurgitation of the bacteria into that bite site. And it just takes very few bacteria introduced into the skin. And then you'll develop what's known as bubonic plague. And so everyone's familiar with that. That's where the bacteria end up in the lymphatics and home to the proximal lymph node. Mm-hmm. You get the large inflamed node full of pus and bacteria. In other instances, you can actually have the bacteria injected straight into the bloodstream, so that bypasses the lymphatics altogether, and you'll develop a form of plague known as septicemic plague. Septicemic plague has a very high fatality rate of approaching 100%.
And the flea can do this?
Yes, the flea can. The bubonic plague is the primary form of plague that will develop from being bitten by a flea, but you can get direct septicemic plague. The third form is pneumonic plague. There's two different forms. There's primary pneumonic plague and secondary pneumonic plague. So secondary pneumonic plague is when the bacteria have entered the body, usually from bubonic or septicemic, from the flea-borne route, and the bacteria circulate in the blood into the lungs. Bacteria love the lungs, and they grow to very high levels in the lungs, and you start coughing. So if I had secondary pneumonic plague, both of you would be close enough to contract primary pneumonic plague. And that's when you inhale the bacteria directly. And so the first organ colonized is the lungs. the bacteria grow to very high levels in the lungs, and you develop this overwhelming pneumonia with complications of septicemia.
Okay.
It's extremely rapid and also very high fatality rate.
So I have to point out that a while ago on TWiM, we did a paper from the Rocky Mountain Laboratory where they traced the genetic changes needed to change Yersinia. So instead of flowing through the flea, tell me if I have this right, it gets stuck and makes a biofilm.
That's right.
Right?
Yeah.
And that was something we did on TWiM a long time ago. And when I was reading one of your papers, we came across it. So it actually evolved to be able to do that. And probably a mutation arose at some point that got it stuck in the flea, and that led to selection of this, right?
Absolutely.
All right, so throughout history, this bacterium has caused lots of plagues or pandemics.
Sure.
Right?
It is, when you think of plague, you think of the plague, which is Yersinia pestis. There's lots of plagues, but the plague is pestis.
So it's killed many millions of people.
Yeah.
And in epidemic form, when there's a lot of people who have been infected, they're coughing, as you say, and they're infecting each other by the aerosol route. Did these start with aerosols or with fleas? Do we know?
Right. So that's actually a really interesting question that that many labs have been trying to work out. And so about a year or so ago, we published a paper in which we identified the earliest point at which Yersinia pestis was competent to cause pneumonic plague, and it was through the acquisition of a small plasmid that carries the gene for a bacterial protease that we study. It's called PLA. But what was interesting is that these variants of PLA had a single amino acid variant, which made PLA less able to allow for bubonic plague to develop. And those are in ancestral strains. But as you evolve towards more modern strains, there's a substitution in that amino acid which enhances bubonic plague. But that substitution doesn't make a difference to cause pneumonic plague, the respiratory form. So we had hypothesized that perhaps pneumonic plague came first. Then in September of last year, there was a wonderful paper published by Rasmussen in Cell. And so what this group had done is that they had been digging up grave sites, skeletons from grave sites from Bronze Age grave sites. And so they were interested in human evolution. And so they were sequencing the DNA recovered from the teeth. And they were getting a lot of this junk DNA that wasn't aligning with human. And so they were just throwing it out. Then someone in the lab said, hey, well, what is this junk DNA? And so they went ahead and just blasted it without a filter. And they found pestis DNA. And so there's a couple of really cool things that came out of this. The first was that it reset when we believe pestis first came into contact with humans about 3,000 years earlier than we had previously thought. But what was especially cool was that all the strains that they were able to isolate of pestis, or the DNA, they all had this gene for the protease, PLA, but they lacked the genes to be transmitted by fleas. So that suggests that perhaps pneumonic plague really was circulating first, or at least not flea-borne transmission was not occurring. And so that sort of really changed the way we've been thinking about how pestis evolved as a pathogen.
So it's interesting that you can talk about ancestral strains. I presume when you get these sequences from the teeth, it's very small pieces, right?
Yeah. Well,
it's not the whole genome. Right?
Well, so actually, this group has been able to isolate the whole genome from two or three of the skeletons. And so they have 95% coverage, which is fantastic.
That's great. That's great. You know, I was thinking about virology and comparing it. In virology, we really can't do a similar study because we don't have anything much older than 100 years.
Yeah.
You know, we have sequences, bits and pieces of sequences, but they don't stick around as well, especially the RNA viruses. They're
very small to begin with, so you don't have a lot to go with.
So the whole idea that pathogens evolve and change to accommodate their hosts and be transmitted more effectively and so forth, we can only speculate in virology. So this is a wonderful example where you have old samples.
Right.
Now, before these Bronze Age specimens were obtained, my understanding is that there are hundreds and hundreds of isolates from all over the world, from recent outbreaks in China, for example, in Asia. How can you use those also to backdate and see what's going on?
Yeah, so that's a great question. So what's really wonderful about using Yersinia as a model to study bacterial evolution is that it has, evolutionarily speaking, emerged very recently. Of course, we've now reset the clock back another 3,000 years, but it's maybe 10 to 20,000 years ago is when it emerged as a distinct species or subspecies from its predecessor, Yersinia pseudotuberculosis. And so that means it's actually relatively clonal. So yes, of course, there's some gene gains and gene losses and accumulation of single nucleotide polymorphisms. But they're not so many, and the strains are not so divergent from one another that it makes it impossible to recreate and trace back these evolutionary processes, at least the ones that were significant. And so we have a great animal model to study mammalian plague. It's actually not even a model. We use mice, but you can use rats, you can use guinea pigs.
Because they're infected normally.
Yeah,
right,
they're infected normally, right? So it's not. So of course we experimentally infect them, and we use the mouse because they're easy to handle, but they're also genetically tractable. So we can look at host genes as well as bacterial genes. But what we can do is we can go back into the strains, modern strains that are isolated from currently infected plague patients, and then we can recreate or retrace back the changes that we find in these ancestral strains or from the skeletons, and make those substitutions and reversions and see which ones were really significant in causing pneumonic or bubonic plague, and which ones were neutral or dispensable for the evolution.
And a similar approach was used to show how the bacterium changed to get stuck in the flea.
That's exactly right. That's exactly right. So what the Hindenbusch lab has been doing at Rocky Mountain Labs for flea, we've been doing for pneumonic plague.
So in a mouse, you get a similar outcome as in people. You get swollen lymph nodes. Can you get aerosol transmission as well?
So that's the one benefit and or drawback of the mouse model. So mice are, they really do faithfully recapitulate what you see in a human. They will get buboes. Of course, you can imagine a mouse lymph node is very small. So, but as the disease enters the terminal phase, you can actually see the mice starting to limp, and they might get, if you've infected them up around here, you'll see it under the armpit. The one drawback with the model is that mice can't cough. So they can't transmit from mouse to mouse. So there is actually no good animal model to study mammal to mammal, direct mammal to mammal transmission.
You need to try ferrets.
You've read my mind. And
if you can, they might not.
Yeah, I mean, I think that's a wonderful thing to do, and I'm actually working on a proposal just to do just that, to establish a ferret model for pneumonic plague.
So even if you take an infected mouse and put it in the same cage, they will not transmit.
No, they will not transmit. So that's actually the benefit, is that each mouse is sort of its own isolated unit, so you can have 5 mice in the same cage and not worry about secondary contamination.
And that probably limits the containment that you need to do. Is BSL-3?
Yeah, it is BSL-3.
Because if it were transmissible to you, you might need to do it in 4 containment.
Well, right? I mean, it could be transmissible to us if we mishandle the mice, if they bite us, or if we are sloppy. Fortunately, everyone in my lab is not sloppy. They'll tell you to pay attention to everybody in the lab. I just vouched for you guys. But we do work in BSL-3 environment, and we wear PAPRs, so these are powered air purifying respirators. Everyone is very highly trained. So, we haven't had any incidents, knock on wood.
So pseudotuberculosis.
Yes.
This can also infect people as well? Where did it get that name?
So Yersinia pseudotuberculosis, it's called that because the original observations of the pathology of the tissues, it looked very much like tuberculosis infection. So pseudotuberculosis forms granuloma-like lesions. It forms these microabscesses. And so the original investigators say, hey, that looks like tuberculosis, but it's not. So let's call it pseudotuberculosis. So pseudotuberculosis is a pathogen of mammals, but it's a mild pathogen. It causes really self-limiting infections.
So it's a gastrointestinal
infection? Right. So it's a fecal-oral transmission.
All right. Yeah. Now, where would you get either Y tuberculosis or pseudotuberculosis or Y pestis nowadays?
So pseudotuberculosis is not so much a problem in the United States. You can find it in Europe, and you can find it in Asia. Generally speaking, though, most people who are infected might not even know that they're infected, or they might have a limited diarrhea and not go to the hospital, and they'll resolve. It's only in the severe cases where you get the rare instance of a dissemination or a systemic event that people might be needed to be hospitalized. So pseudotuberculosis is still circulating. You can find it. You'll often get it from eating undercooked pork. So your pigs, you got to make sure you really cook it well.
Is it in rodents in the wild as well? Or not
yeah,
so
but
much?
it's not. But it's not something that is routinely checked.
So where do the pigs get it? They're presumably on a farm, right?
Yeah.
Where are they getting it from?
It's just out in the wild.
Okay,
All
right.
But pestis, on the other hand, is endemic on all the continents except for Australia and Antarctica. We have it in the United States. We have maybe 8 to 15 cases of plague a year. 2005 was actually a high number. We had 15.
Where are they usually in the Southwest?
So it's in the Southwest, usually west of the Rockies. And so in the United States, people usually get infected because they come into contact with infected rodents or fleas, or maybe their house pets. So a great example is the strain that we work with was isolated from a woman who developed pneumonic plague because she caught it from her cat. So the cat essentially coughed in her face. The cat had caught a dead or dying rodent, ate it, got plague, coughed in her face, and then she developed pneumonic plague as well.
Pneumonic, okay.
Yeah.
Did she survive?
She unfortunately did not.
Because you probably don't pick it up quick enough, right?
Well, that's the problem, is that the disease is very rapidly progressing, particularly pneumonic plague. It's about 3 to 4 days from infection to death. So you have to be very astute in diagnosis, because if you don't, then if you miss that window in which antibiotics can be effective, then you're done.
And with so few cases, it's hard to recognize it, right?
Yeah.
Maybe in the Southwest, physicians need to be
pay
a
more
little bit
attention.
aware, but certainly not in the Northeast. Are there never any cases in the Northeast?
No, they're usually imported. So there were a couple of individuals a few years ago who did develop plague, and they were in New York City. And so, of course, the first concern was, is plague actually circulating in New York City? Right. And it turns out that they were visiting the Southwest.
So it's a select agent because of the fatality and the aerosol transmission, right?
And the fact that it actually has been weaponized and used as a bioweapon in the past.
So when was it used? Tell us.
So it was used centuries ago by China. The Chinese has, but actually more recently by the Japanese against the Chinese. They actually released infected fleas into a Chinese population, and there was an outbreak of bubonic plague. Yeah.
There are also stories of armies throwing infected animals over the walls, right?
Yeah.
And spreading
infected
it that
cows,
way.
right? Yeah.
And in modern times, it's been weaponized as well.
It has. So the Soviet Union had weaponized Yersinia pestis, made it antibiotic resistant, which is quite scary, because if a multi-drug resistant strain of Yersinia pestis were to start circulating, then we'd have severe pandemics.
So you must get tons of money from the government to work on this.
One would hope. You tell study section, they're reviewing my grant tomorrow.
So, seriously, what is the situation? This is a biodefense meeting. It's also got emerging pathogens there. But biodefense part, is it, and you can also speak to this, Rebekah, is it well supported, or is it just something that we talk about but don't really support research in the area?
Yeah. Well, so at least from the perspective of bacterial pathogens, I think after the anthrax letters attacks, there was this big push by the U.S. government to throw a lot of money towards biodefense. And so one of the outcomes was forming these regional centers of excellence for biodefense. So I was part of the Midwest Center when I was in St. Louis, and then the Great Lakes Center when I moved to Northwestern. So those went through 2 rounds of funding, and they were a great resource for starting new projects, and we were funded through that. But then they were not renewed for a 3rd term, and so the center shut down. And so now, at least for basic research, our primary money source is NIH standard NIAID funding mechanisms. There, for people who are doing maybe more applied or clinical research, there's DITRA, there's DOE, and so on.
What is your experience, Rebekah?
My experience, when West Nile virus first came here, I think it was a wake-up call that there wasn't a lot of the necessary infrastructure out there to handle that. So that created a wave of funding and research that lasted, a decade just about. And then since then, I think that's for something like RIFT, there's more, like you mentioned, DITRA and DOD would have interest, and I think this is a USDA concern as well. So I think there's some diverse funding opportunities for that.
Yeah. Unfortunately, one of the issues that at least we in the pestis field have to deal with is because there are not a lot of cases that occur in the United States anymore. And in fact, worldwide, there may be several thousand cases a year, but it's not on the level of malaria or tuberculosis or some viral infections. Sometimes we get comments that say, well, nobody gets plague anymore, so why should we continue to fund the research? And unfortunately, I think that's a little misguided because, of course, in such as what we had with Ebola or maybe what's going on with Zika, we have to be prepared. But also, I think there's a lot of basic biology we can learn about host pathogen interactions, host immunity, and infectious disease biology by studying Yersinia pestis. That works as an evolution even is a great model.
All these things are right. In fact, when the government says here's money to work on a pathogen that's important for biodefense, in fact, you learn a lot of basic biology from doing that, and that's a great outcome, even if you don't develop a vaccine or an antiviral. Polio hardly infects anyone anymore, yet we can still learn about pathogenesis by studying these model organisms. So I would say, all you have to do is write a good grant proposal, right? And if you could make the case that it's important to study your organism, then it should be supported. And so even when the biodefense money gets very low, as you said, you go through the normal mechanisms and you have to make a case. Okay, there's not a lot of Yersinia in the U.S., but it still can teach us, right? I wanted to go back to one thing you said earlier about this protease that you think is important. And in fact, in one of your papers, you traced how you thought pseudotuberculosis became pestis by acquiring a plasmid that has this protease.
That's right.
How did you do that?
So it's well established that this plasmid, it's called PPCP1, but we just call it the PLA plasmid, is found in almost all modern strains of Yersinia pestis. And indeed, it's required to cause, the protease is required to cause both bubonic and pneumonic plague.
You can tell that by getting rid of it and putting it in mice.
That's right. So if we do a gene knockout, the bacteria are highly attenuated, if not avirulent. But there aren't any strains of Yersinia pseudotuberculosis that carry this plasmid. So it's really a night and day difference. So this was an acquisition event that had to occur. But so what we wanted to do was we wanted to test whether these ancestral strains, and some of which don't have the plasmid, were they competent to cause pneumonic plague? And we found that they were, but only if they had the plasmid. And then work, of course, that from the archaeological evidence showed that this was one of the very first acquisition events that occurred in following the emergence of Yersinia pestis from pseudotuberculosis.
So the Bronze Age sequencing did not reveal the presence of
No,
this plasmid?
all the strains had the plasmid.
Okay.
Oh, they all had the plasmid. So what's interesting, though, is that putting this protease into pseudotuberculosis does not necessarily endow pseudotuberculosis with the ability to cause plague.
So you were actually allowed to do that experiment?
Well, it depends on who you ask. So this was actually originally done by the French, by Elizabeth Carniel's group at the Pasteur Institute. And so they actually showed that this was not, you didn't make a super plague by doing this. And so some other groups have since done it. And since it's now been shown that that's not, you're not doing necessarily gain of function type research, people can do.
How long ago did the French do that experiment?
Oh, I think that was 2003, 2004. It was certainly last decade.
So that's before the current climate on pauses and so forth against what are thought to be dangerous.
So we have not been, I think, affected necessarily by this pause in gain of function research. I think that's been mostly directed at the influenza community.
And a little bit of coronaviruses as well.
But one of the things that we need to do in order to study pathogen evolution is we need to take inactivated virulence, hypothesized virulence factors that have been activated and reactivate them, restore them to see whether that loss was consequential in the evolution. And so fortunately we do all this work in the BSL-3, so if we do create a more virulent pathogen, it's contained. Right. All of this has to be pre-approved.
I think that's the way the process should be, rather than banning outright certain types of experiments. Just say, look, you make a proposal, we'll review it, we'll tell you what kind of containment you need, and if you can do it, you do it. Because you never know what's going to happen when you do an experiment in science, right? You can't predict.
Well, and that's one of the things that I teach, is that it's gain of function, but is it gain of function of concern? And I think that's the distinguishing point, is if there is a concern, then you might need to investigate whether that should be done. But because a lot of what we do is gain of function. Of course, that's how we must do our science.
And I must say, you do not want to make the acronym for gain of function of concern. Not on this show, because we are a clean, family-rated show. But you can do the experiment in your head and see what it comes out to be. You know what I'm getting at? In 1 minute or less, what does the protease do to make the bacterium more virulent?
So the protease was originally identified as a plasminogen activator. So it's called PLA because it activates plasminogen. And so we've recently shown, and we're about to submit a paper that shows that indeed the conversion of plasminogen to plasmin by this protease is essential for the bacteria to cause pneumonic plague. If you take away PLA or you take away plasminogen, you are dead in the water and you can't cause pneumonia anymore.
Wonderful.
Yeah.
This is a really clean phenotype.
It really is.
You don't often get that lucky.
Yeah.
Right.
No, the two deletions, they phenocopy one another.
All right.
Yeah.
Is anyone brave enough to ask a question before we wrap up? We have 2 minutes. No? Yes.
Hi, I'm John Boyle from the University of Pittsburgh. How, in terms of mosquitoes and different species, why they do and do not transmit different viruses, is the mechanisms or the differences primarily physical, that the viruses aren't able to cross, for example, out of the midgut, or is the mosquito doing something as well? Like how many, sort of thinking of physical barriers versus maybe immunological barriers in these different mosquito species?
That's a good question. There's both. Mosquitoes have immune systems too. They're not like ours, but they are mounting a defense against these viruses that are invading, and so that is something that is going on as well. And then sometimes there may not be the right receptor there for the virus to get in, or there's a lot of different things going on.
And in your work, do you sort of think about about whether certain viruses could evolve to be transmitted by mosquitoes and whether those mechanisms are more or less likely to be overcome?
Absolutely. One example would be chikungunya virus. Developed a mutation, just a naturally occurring mutation on the E protein that allowed it to be more infectious to Aedes albopictus mosquitoes. And so then, this is one example of just, something that was a fitness benefit to the virus, that then it persisted. And so it was able to move into a new vector.
All right. Thank you. That'll do it for this episode of TWiM. We have to move out of this room. You can find TWiM at iTunes, at microbeworld.org, and at microbe.tv/TWiM. And we love getting questions and comments. We have a great interactivity with our listeners. You can send them to twim@microbe.tv. I want to thank my guests for joining me today from the University of Colorado, Rebekah Kading. Thank you so much.
Thank you.
Hope you enjoyed it. And from Northwestern University Medical Center, Wyndham, Lathem. Thank you. Is that right?
Lathem, that's right.
I got it. Amazing. Well, I had two choices, right? Could go either way. I'm Vincent. I meant to say, you're on Twitter.
That's right.
W. Lathem.
That's right. Follow me.
Follow him on Twitter.
Right.
You're not on Twitter
I'm
yet.
not on Twitter.
You should get a Twitter account. I could plug it and get you thousands and thousands of followers. And I'm PROFVRR on Twitter. You can also find me at virology.ws. And of course, I'm Vincent Racaniello. I would like to thank the American Society for Microbiology for allowing us to do TWiM, and the meeting organizers here at Biodefense, including Katie Bozio, who's sitting there listening. Thank you very much for having us. And I want to thank our producer today, Chris Kandayan, who's sitting over there with the headphones. Thank you, Chris. And Ray Ortega, who does a lot of behind-the-scenes technical help for us at TWiM. The music you hear at the beginning and end of TWiM is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.